I'm going to talk about one aspect of the work that we have been doing at the Chinese university. And like Veronica, I have a strong link with ANU. Actually, I was graduated from a PhD from ANU long time ago, right? But anyway, I've been visit Australia many times. And today's presentation, I will concentrate on one anti-cancer compound called anti-tumor oxime, using the model human neuroblastoma cells. And basically, in the next 20 minutes of my presentation, I will go through a number of things. Like today's second speaker, she had introduced to you the human neuroblastoma, so I will almost skip this session. And then I will talk about the anti-cancer compound. It's a natural product called anti-tumor oxime, abbreviated as I3M. And we go through the data on its anti-tumor effect on human neuroblastoma. And then we try to look at the mechanism by which the I3M can exert the anti-tumor effect on neuroblastoma. And finally, we would like to have some conclusions and the future outlook for research. And so again, what is neuroblastoma? As mentioned from the previous speaker, this is an extracranial, that means outside the brain. This is a solid tumor derived from the so-called primitive cells. That means the neural crest cells in the embryo. And basically, this will arise in the sympathetic nervous system. And this is the most common and most deadly solid tumor of childhood. And it accounts for about 8 to 10% of all childhood cancer, and also about 15% of all childhood cancer-related death. And what are the symptoms? And you can see the cancer originates mainly in the sympathetic nervous tissue. Most of them come from the adrenal gland. But however, a tumor in the abdomen, you can see here, basically, there's a swollen belly, and this will cause constipation. And if a tumor in the chest in the infant will cause difficulty in breathing. And finally, if the tumor found in the bones around the eyes or orbits, you can see very distinct bruising or inflammation. So, what are the etiology of neuroblastoma? It's not well understood. And basically, a number of risk factors has been proposed, like, for example, certain occupation, exposure to chemicals in certain industry, smoking, alcohol consumption, or using certain hormones and fertility drugs. And also, this has been associated with maternal use of the hair dye and use of medicinal drugs during the pregnancy. However, a number of protective factors for neuroblastoma development is the use of what? Supplements, the folic acid supplements, and the vitamins. But anyway, let us look at the molecular pathogenesis of neuroblastoma before we talk about the anti-tumor mechanisms. And basically, again, mentioned by the previous speaker, in about 20 to 30% of all neuroblastoma, there's a so-called MYC amplification. This oncogene becomes amplified. And this plays a very important role because this is associated with high-risk neuroblastoma and also poor prognosis and survival. And again, there will be chromosome abnormalities seen in neuroblastoma patients. For example, there's a chromosome deletion in chromosome 1 and also 11. And there are mutations being observed in this so-called pair-like homeobox gene and also the so-called anaplastic lymphoma kinase gene. And these two genes are known to be associated with neuronal cell development. And also, as usual, usually many cancers are associated with disruption, aberrations in the cell cycle division, and also apoptosis process and other signaling pathways. So basically, the current treatment for neuroblastoma depends on the types. And according to the International Neuroblastoma High-Risk Risk Group Classification, there are 3 different types. And for the low risk, for example, the cure rate is more than 90% using surgery alone. And for intermediate risk, all this without MYC amplification, one can deal with surgery and chemotherapy and have a very high cure rate. However, for the high-risk one, which will be the focus of this study, and you can see here, despite the multi-modality therapy using in different treatments, the cure rate is only about 30%. So there's a pressing need, an urgent need to look for alternatives which can deal with the high-risk neuroblastoma. And this sets the stage for our research. And we try to look for natural compounds. And this is one of them called indirubin-3 oxime. And you can see here, this is a cell-permeable derivative of indirubin. And this indirubin, you can see here, the basic structure is an alkaloid. It's a wheat base derived from natural products. And basically, indirubin is an indo-alkaloid, and it's isolated from dry root. This is the dry root, all right? And which is from the different medicinal indigo plants. And you can see this is the I3M. This is being modified from indirubin. And from here, you can see the oxygen is being replaced by NOH. And this compound is available commercially, have a higher solubility, and also a higher activity. So based on this background information, we look at the literature. What are the biological and pharmacological activity of this compound? And as shown here in this slide, it can inhibit the CDK, the cyclic-dependent kinase, and the glycogen synthase kinase, and also have a potent anti-inflammatory activity. In fact, the indirubin have been used in the Chinese medicine for treatment of certain infection. For example, viral infection, hepatitis, influenza, or even SARS virus infection. And also for certain chronic inflammatory disease like ulcer. So have a well-known antiviral activity, but in our study, we focus on the so-called anti-tumor activity, anti-angiogenic activity, as well as the anti-tumor mechanism. So in our assay, we use the so-called MTT assay, calorimetric assay, and looking at the ability of this I3N compound to inhibit the growth of different neuroblastoma cell lines. And you can see here there are 3 different cell lines being used. 2 of them, LAM1 and SK cell lines, they have an MYC amplification. That means they belong to the high-risk neuroblastoma. And you can see, interestingly, these have a very high potency, I3M, on this high-risk neuroblastoma, where SH neuroblastoma cell have only one single copy of MYC, have a relatively lower IC50, the inhibitory 50 concentration. So we focus on the ability of I3M to have an anti-tumor effect on the high-risk neuroblastoma LAM1 cell. It's important a compound, it can be used for clinically, right? It should have a low toxicity to the normal cell. So we try different normal cells. One is the counterpart, right? It's the so-called SD-RAC cortical neuron, primary neuron, isolated from the RAC as a control. And also there are cell lines like human embryonic kidney cell and the so-called WRL cell is the hepatocyte-like cell. And you can see it have a very low toxicity in this primary cell or non-transformed cells. And one of the possible mechanisms why have I3M have anti-tumor effect, and we try to look at the ability of I3M to induce apoptosis using an ELISA assay which measures the DNA fragmentation. However, we failed to detect any induction of apoptosis. As a positive control, using a chemotherapeutic drug, doxorubicin, have a high activity for the induction of apoptosis in the cancer cell. So, induction of apoptosis is not one of the mechanisms. How about cell cycle arrest? We try to do cell cycle arrest, and you can see here, I3M can arrest the cell cycle of LAM1 cell. And you can see here, it's basically, here is the G1 cell arrest with a simultaneous decrease in the cells at the S phase. And we look at the different cell cycle regulatory protein, the CDKI, the P27 inhibitor of CDK, and also CDK2 and cyclin E. And you can see here there's an increase in the CDKI but a decrease in the CDK2 and cyclin E, which is known to be associated with the cell cycle progression from G1 to S phase. And this is the result of the Western blot. So again, an interesting phenomenon is the association between mitochondrial biogenesis and function and cell cycle. And from the literature, you can see that using the human HeLa cell, the spherical carcinoma cell, the expression of a factor called mitochondrial termination, transcription termination factor 4. A reduction in this transcription factor is associated with cell cycle arrest. Whereas in other model animals, for example, Drosophila, the fruit fly, the mutation in the cytochrome oxidase subunit V alpha, there's a reduction in ATP production, and then there's a cell cycle arrest. And similarly, the reduction in the PDSW is a subunit of the mitochondrial complex 1. Again, you can see that there will be an increase in the reactive oxygen species production, and with a decrease in cyclin E and CDKI, which is increased. So all this will have a link up to the mitochondrial biogenesis and function, as well as the cell cycle progression. Based on this one, we studied the mechanism. For example, transcriptional factors like ERR, estrogen-related receptor alpha and gamma. And these are constitutively expressed active nuclear hormone receptors. You can see here it will bind to the DNA, and then in the presence of so-called co-activator, PGC1, that is the peroxisome proliferator activated receptor gamma co-activator, 1 alpha and 1 beta. You can see this will cause the activation of gene expression. So this control a number of gene expression involved in mitochondrial biogenesis and oxidative phosphorylation. So we look at the ability of I3M to affect the gene expression of ERR, gamma, and PGC1 beta. And you can see from the Western blot, there's a clear distinction. There's a down-regulation of ERR, gamma, and down-regulation of PGC1 beta. And this is the result of 3 experiments and so on. And similarly, we look at the gene expression of so-called mitochondrial regulated genes. For example, this is mitochondrial transcription factor A, and also the ATP synthase, which is involved ATP production, and also in the antioxidant enzyme, superoxide dismutase. You can see here, treatment with I3M for neuroblastoma, this will cause the reduction in these transcriptional factors and also certain enzymes, which is involved in mitochondrial function. So we look at the mitochondrial function, and especially the I3M cause so-called mitochondrial dysfunction. For example, there's a reduction in mitochondrial mass, as well as there's a reduction or depolarization of the mitochondrial membrane, as well as we measure the reactive oxygen species, especially the superoxide anion. There's an increase in ROS, whereas there's a decrease in mitochondrial mass and the membrane potential. So to summarize the result, right? So we look at the effect of this anti-cancer compound on human neuroblastoma. And we found that it will downregulate the ERR gamma and PGC1 beta. These 2 genes are involved in the regulation of mitochondrial biogenesis and oxidative phosphorylation. And therefore, we will have found that I3M can cause a reduction in the mitochondrial mass as well as the mitochondrial membrane potential, whereas it can increase ROS production. And somehow, although the link is not clear, we observe there's a reduction in the CDK2 and cyclin E, which is shown to be associated with the arrest at the cell cycle. And also there will be increase in P27, which is a CTK inhibitor. And this will lead to the cell cycle arrest at the G0 and G0 C1 phase, and finally this is a cell growth inhibition. So this is the anti-tumor effect. And lastly, we have performed a series of experiments, and to cut this long story short, we just summarize the result here. We found that I3M also have anti-angiogenic activity on using the human microvascular endothelial cell. And you can see here, treatment of the HMEC1 cell with I3M. This will cause a reduction or expression in the angiogenic factors, like the angiopoietin-1, which is known to be stabilize the blood vessel, and the matrix metalloproteinases 2, which is involved in the degradation of the basement membrane. So there's a reduction in angiogenic factors, and we look at the signaling pathway, and we found that there will be reduction in the activated form of the VEGF R2, that means vascular endothelial growth factor receptor 2. And then subsequently, we look at the signaling pathway, and there's a reduction in the so-called PAKT pathway, as well as the MEK kinase pathway, MEK, ERK, etc. Right? And finally, we observe there's a suppression of endothelial cell proliferation, and also there's a reduction in the migration of the endothelial cell, as well as in the capillary formation. All this will be reduced in vitro. And therefore, we observe the ability of I3M to inhibit angiogenesis in vitro. And finally, it's the last slide, is that talking about the I3M, the ability to reduce the angiogenesis in vivo. Basically, we inject I3M together with the VEGF and so on. And then this is being introduced into the subcutaneously into the bouncy maps, and with the matrix gel. And then after 7 days, we isolate the matrix gel plug, and you can see here the one without I3M have a vascularization, and we measure hemoglobin content and quantify. And you can see here, I3M can actually reduce the angiogenesis in vivo. Lastly is the conclusion. What we have found so far, I3M is a potential therapeutic agent for the high-risk neuroblastoma, like Leung cell with MF amplification. And I3M possesses also potent anti-angiogenic activity, both in vitro and in vivo. And however, currently we are doing the in vivo study of I3M in animal model to make sure it has anti-tumor efficacy and also have a very low acute or chronic toxicity. This is still underway. And finally, hopefully I3M can be used as an agent, combination therapy with the conventional anti-chemotherapeutic drug, and to reduce the dose being used. So that's the end of my presentation, and these are my collaborators and my former PhD students, and also Professor N.K. Mek and Professor N.S. Wong at the Hong Kong Baptist University. So that's the Chinese University, and you're welcome to visit me, right? Thank you for your kind attention.
Okay, any questions?
Yeah, yes, please.
So, has this been tried for the resistances, I3M?
For what?
For chemotherapeutic resistances?
It hasn't been tried because the original compound, indirubin, have been used, right? But this one is a modified one at a higher solubility and higher CDK inhibitor activity. So in the literature or in the animal model, we haven't found any data.
And when you see that it has anti-cancer activity in vitro, so after treatment there are some cells which are left over. So in the consequent facade, if you go with them, so will this be affecting those cells which are the residual cells after treatment?
Yeah, the residual cell.
Yeah.
What do you mean by residual cell? Because we are using the cell line.
So residuals in non-respondent cells. So will it affect again the same I3M at the same concentration, or you have
we use the same concentration or even higher concentration from the normal cell. It apparently is not toxic to the normal cell or even the primary neurons and so on as a counterpart.
And is there any preventive effect of it? I mean, before implantation of the tumor, and if you see any preventive effect?
Preventive effect.
Yeah.
We haven't tried because we try to use it as a therapeutic agent rather than a prophylactic agent. Right? Thank you very much for your suggestion. Yes, please.
I also graduated from AU.
Oh, yeah.
it's because it's your compound seems like it have some effect on mitochondria. And so when you do the apoptosis, I think you may be better to detect, try to like detect like BCL2 or CAR.
Yeah, we have done that before. And usually, first thing we look at the ability of the compound to induce apoptosis using a variety of assays. For example, DNA ladder, DNA fragmentation, the tunnel assay, the ELISA, and so on. And then subsequently, we try to look at the BCL2 expression, cascade activation. So far, using this compound, we failed to induce any apoptosis. But on the other hand, from the same dry root, we isolate other alkaloids. They are able to reduce apoptosis. So it's a clear differential effect, right?